Okay, good morning everyone. Thanks to everyone for coming in so early and being here for the participant presentations, which is going to be our first part of the session this morning. Basically we have three people that have been chosen from the abstracts to give a talk here. I would now introduce our second speaker this morning, who by complete coincidence is also from the European Institute for Oncology in Milan. We did not receive any donations. And her name is Roberta Noberini. She is in the lab of Tiziana Bonaldi, and she's going to be talking about MS-based analysis of histone PTMs in clinical samples. Looking forward to that.
Okay, so good morning, everyone, and thanks to the organizers for the opportunity to present here my work on histone post-translational modifications in clinical samples, and particularly breast cancer samples. So, as you all know, histones are the protein portion of chromatin and are modified with a number of post-translational modifications that define a very complex code that is functional to regulate nuclear functions and gene expression, ultimately defining cell fate. This code has been plays a crucial role not only in physiological conditions, but also have been linked. Aberrations of such a code have been linked with a number of different diseases, including cancer, which is the focus of our lab. Here, just to give you an idea of how relevant studying epigenetic features and histone PTMs in particular in cancer samples, here is the list of all the modifications of all the histone PTMs that have been found aberrant in different ways in different cancers on histone H3 only. And one example of really nice findings that were obtained in cancer studying histone PTMs is shown here by these two papers that identified histone PTM combinations as prognostic factors in prostate cancer, breast cancers, but also many others. However, how it is exemplified by these two papers, most of the studies that have been done so far on histone PTMs in the clinic have been carried out using immunohistochemistry. And I don't think I have to convince this audience how better suited mass spectrometry is for PTMs analysis, and particularly histone PTMs analysis, thanks to the fact that it's much more comprehensive, unbiased, quantitative, and at least up to a certain point allows detecting combinations of modifications that can give insights on PTM's crosstalks. So with the idea of merging the power of mass spectrometry-based analysis of histone PTMs with the potential offered by the clinic, we developed, and we have focused in the lab in the last few years on developing different protocols tailored for the analysis of clinical samples and primary samples in general, which include primary cells, frozen tissues, and FFP tissues, which are formalin-fixed paraffin-embedded tissues. We've heard about them yesterday, and I just wanted to remind you that these are particularly interesting for us because this is the storage method of choice for most clinical samples in hospital biobanks, and as such are an invaluable source of clinical information. And for this type of samples, we even developed, we implemented the protocol to be able to analyze laser microdissected cell populations and tumor populations. And we've shown how analyzing these subpopulations allows to detect bigger changes, as one might expect, compared to the original method. And once we have set up all these proteomics method for the analysis of histone PTMs, we are now trying to apply them to different clinical projects or more clinically oriented projects. One of our main focus is breast cancer, which is still the leading cause of death for women worldwide and can be divided into 4 subtypes that are defined based on the expression of 3 molecular targets that also guide the choice for the treatment. Among these subtypes, what we are focusing on is the triple negative subtype that is defined based on the fact that it does not have any molecular targets and therefore does not have any targeted treatments in addition to chemotherapy, and its prognosis is therefore particularly low. In this context, we are now carrying out the profiling of histone PTMs on different sets of samples, and I'm going to show you just a few examples of what we are doing. But the general idea is that aberrant histone PTMs, or more in general changes in histone PTMs levels, can provide on the one side information about possible epigenetic biomarkers for patient stratification, but on the other side also suggest molecular mechanisms and potential novel epigenetic therapeutic targets. And this is thanks to the fact that aberrations and changes in levels of histone PTMs are usually linked with the abnormal activity of histone modifying enzymes. And because a lot of interest has grown in the last years about epigenetic therapy, especially for cancer, a lot of epigenetic drugs targeting these enzymes have been already developed and could be ready to use if we identify these potential targets. So here is one example of the kind of results that we get from our PTMs profiling. It was sort of a proof of concept experiment where we compared these histone post-translational modifications across many breast cancer samples belonging to different subtypes. And the idea was, well, can we see epigenetic changes in different subtypes? And the answer is yes. Well, the coloring refers to different relative levels of each of the peptides that we obtained using a histone-focused super-SILEC approach. And as you can see, there are a number of significant changes that differentiate the different subtypes. And I'm not going to go into the details of the specific PTMs that we found, but I just wanted to mention that we are trying to define a signature that would distinguish triple negative compared to the other subtypes. And when we looked deeper into the triple negative subgroup, we did see a certain level of heterogeneity even within the group, which is somewhat not surprising given that this is still a very heterogeneous set of samples, given the fact that they are defined by the lack of the targets that characterize the other subtypes. And this is something that we are going to expand and we need to expand, but the finding that there are differences is promising because it might suggest that we could be able to define epigenetic defined subgroups that may be even characterized by specific epigenetic targets. And we also analyzed these sets of samples from another point of view, meaning based on the fact that they did or did not relapse 3 years after chemotherapy. And even though these sets of samples is rather limited, and without taking into account this heterogeneity, which might be a very important factor, we were able to detect some significant differences among these two groups. And this is something that we're going to investigate further by, of course, first validating it on a larger cohort of patients, both with the idea of using this as predictive biomarkers for response to therapy, but also suggestive of potential targets. And I would like to conclude with something that we are really interested in now, but it's that we are starting to do now, which means analyzing histone PTMs by mass spectrometry on circulating nucleosomes. So what I've showed you so far has been done in tissue, and it can be very useful for finding novel mechanisms and even for biomarkers. But we've heard yesterday how circulating biomarkers would be much preferable. And circulating nucleosomes are a rather exciting area of research that involves the analysis of these nucleosomes that are released into the blood from after cell death and apoptosis, are usually higher in cancer tissues, in cancer patients, and usually carry a signature of the cells from which they are derived. But usually, as it is often the case for PTMs, histone PTMs, they are analyzed by ELISA assays and, in general, antibody-based methods. What we're trying to do, and we've done very preliminary experiments, but really promising ones, is to use a protocol that's been published recently, pretty straightforward, that allows to isolate histones for the analysis of PTMs by mass spectrometry. And we are adapting it to rather limited amounts of sorting material, which would be compatible with the amounts that are available in biobanks. And remarkably, we were able to identify many of the most common PTMs that we usually profile. And so this is actually something very interesting that we are focusing on. So I will stop here, and I will end by acknowledging Tiziana Bonaldi and all the Bonaldi group and our collaborators from the IO Hospital. And thank you for your attention.
Okay, so as I mentioned, we'll have questions at the end. So that gives me time to introduce our final speaker from the participants, and that's Emanuele Alpi. He's a senior technical officer and bioinformatician at the EBI UniProt development team. And he is going to talk about integrating genomics and proteomics in the UniProt resource.
All right, good morning, everyone. So thanks the organizer for letting me present here some of the work that we do in the UniProt resource. So what happened 16 years ago is that these three institutions in these different parts of the world decided to get together, to merge and integrate their resources into the UniProt resource by creating the UniProt consortium. So UniProt stands for the Universal Protein Resource. And the aim here is to provide the highest quality, most comprehensive, most thoroughly annotated, fully public protein resource concerning everything related to proteins. So I'm just quickly jumping into where a simplified version of a simplified view of the sequence data flow into UniProt. And I want to highlight on a couple of things here on this slide. So first of all, the two main sections of UniProt. One is called the UniProtKB. KB stands for knowledge base, okay, Tremble. The other one is the reviewed one, is UniProt knowledge base SwissProt. And upon annotation, manual curation done by curators, so physical person, entries can flow from the unreviewed part to the reviewed section. But unreviewed doesn't mean at all bad in any ways. It's just that it didn't have manual curation for the moment. It's just that, okay? And I want to highlight this point by letting you understand here where our bulk of the sequences come from. So you can see that we import basically amino acidic sequence. They are coded here in color-coded in green. And most of them come from the ENA, which is part of the INSDC consortium. So, but we do import also from many other resources. As I said, this is a simplified version. So suppose we do also import from Ensemble. For us, Ensemble, for instance, considering human represents the reference genome. They are the ones who tell us what are the coding sequences. So suppose they come up with a new additional gene that even for human was not known before, or a correction of a previous gene or anything like that. Initially, the new sequence will flow into tremble, okay? All these sequences, as you see, flow into tremble. So if you a priori exclude the tremble entries from your analysis, you might be losing relevant information, even for human, which is the most highly annotated and studied species. We do have some small-scale direct amino acidic sequencing, manual sequencing submissions that upon annotation and curation, again by a curator, can flow directly into the SwissProt part. But the vast majority initially flows into the tremble section. So take-home message here, excluding tremble a priori, yeah, you can do whatever you want, but at your own risk. Then again, we have a website. You can find it here. We do provide lots of things on this website. We have color coding, depending if you are in the UniProt knowledge base section or color codes for these other sections. And we do provide lots of capabilities from the website. These are the numbers. For the current publicly release, we do have a release number. This is the version, so it's year underscore and 2 digits. We provide this release number in many places. As you can see, there's quite a nice amount of sequences in the tremble section. So I want to highlight here this word, again, to stress that there's nothing wrong with tremble sequences. They are just awaiting full manual annotation, okay? This is, again, to give you some more details about the two sections, but I just want to highlight two things here, that we do lots of automatic annotation computationally for all the tremble entries. It's quite a high number, so it needs a lot of computation power, and it's expert-led automatic annotation. It's not blind automatic annotation. And even though that sequence redundancy is allowed in the tremble section, we do remove redundancy at the level of entire proteomes against each other, analyzed in closely related taxonomy space. And you can see here from this big bump down, the moment when we started introducing this sequence redundancy for removal at the proteome level for bacterial world. And we did this because we were experiencing very exponential growth of the database, so we tried to reduce this. And it's growing still quite a lot anyways, but we have to also expand the redundancy removal also to other kingdoms. So again, highlight on the await word there, and the same take-home message as before. What I want to focus in this presentation actually is what we provide in terms of proteomics and genomics to this RESTful API, which is available at this URL here, and this is a snapshot. So we do a lot of mappings from large-scale projects. And we decided, these mappings don't flow into the classical UniProt flat files, like the .dat files, which you might be using, or the XML files. They are reported on the FTP, obviously, in tabular formats and in many other formats, but they are not in the UniProt flat file. So we decided we want to expose this more to the users. We came up with this RESTful API, and there we provide in a programmatic way. So people with a little bit of programming experience, but not that much, can start using it. And we published this last year, and we have examples, release notes, and contacts, so anything that for bug fixes and features which are not there, you can contact us. There's active development. We are adding content to this API every month. So, also nice, this sentence here, I don't know if you can read it, but for the moment we don't limit user requests, so you can query this API even in a high throughput. Obviously, you cannot get everything in 1 second. And if you try to bring down our services, we have a way to defend ourselves and to let you know what is the proper way to query in the API. But we don't limit it for the moment, just because we want to see how much, what are all the different use case, for the users. I just want to highlight here two services, which are the proteomics, that I will talk a little bit, and also the genomics one, like for instance, the genomic coordinates, there you go, and the variation data that we integrate. But also the classical UniProt features are available from here. So suppose that a couple of persons here in this audience are not well satisfied by the XML files that we produce, why don't they try to give a go at this RESTful API to see if everything is in there, or if something is missing, we can try to add it. So this is why we did it, and I already said this. I just want to focus on the coordinate service. So, because sometimes I've heard that, well, I'm not using UniProt, I'm going to other database, that's super fine, and the reason is because I'm not finding genomic coordinates. We do have genomic coordinates, and we do expose them to the public, and they are all available here. Same goes for the proteomes which are made redundant by the procedure which I've shown before. They are all available here. We don't delete anything, obviously. And we can also reinstate redundant proteomes into the UniProt knowledge base upon request, obviously. So this is just a quick snapshot of one of the ways in which proteomics data can flow into UniProt. It's not the only way. And here I won't go into the details. I just want to say briefly that if we have a proteomics repositories out there storing and providing identification results, most of them are bottom-up, but also top-down proteomics is available. We can try to map this data to our sequences, okay, by doing sort of a very, very simple pipeline here. And so here the aim is to map a mapping service. There's no aim to reproduce what the repositories are doing, nor to provide the high level of detail which DMS proteomics repositories are providing. Then we try to map this proteomics data. Why? Because then we want to bring the protein existence values that we have for each protein to the value of 1. Okay, when we try to map this proteomics data, evidence at the protein level. We think the NMS proteomics nicely fits with evidence at the protein level with respect to the other categories there. So once we do this mapping, we provide them on the FTP with all the details of how we do it, and everything is there. Then this flows into the RESTful API, which I've shown you. And it goes also on the website via this feature viewer. This is an open source software which is, many groups inside the campus and also collaborators outside the campus are using on their own, because it's a very nice way to have something already built that can interface directly to the visualization part of UniProt. And the corresponding papers are there. So I'm not sure how many of you know, once you land on a UniProt webpage, there are these clickable items here on the left. And the entry one will be the one selected by default. But you can click also on the others, one of which is this feature viewer. Upon clicking on that, there will be some graphical display. This is not an up-to-date snapshot anyways, showing many feature viewer, and you can zoom in, zoom out in the sequence, and we are always developing this. There's also lots of active development going here. 3D integration of 3D structure together with this feature will come very soon. This, for the moment, is working only for canonical sequences. It will be extended soon. So you can see that there is a proteomics track there. You can expand it, and you can get to see the details of the mappings of these peptides, positional, and where do they come from, which repositories. Same goes for the variation part. I didn't talk yet about the genomic part. So we do map a lot of variation for large scale projects. They are all reported here also in a graphical way. And also color coding, so you can fish out, for instance, there's some disease-related variant, and by clicking on it, you get the details of the variant itself and the corresponding disease. So querying the RESTful API in terms of proteomics and variants and genomic coordinates. So for proteomics, you will, by querying it, among the details that you will get always the positional features, the positional details, within the protein sequence. So this human peptide here starts at this amino acidic position and ends here. Proteomics can be queried across all the covered species, which are 11 so far. And by the genomic coordinates, you can also get to know that this peptide here is sitting on chromosome 9 and starting at this nucleotide position, ending at this nucleotide position. And you can see by the numbers here that we are on the reverse strand. And obviously, the genomic coordinates support intron and exon spanning. So quickly on variants, we covered 32 species so far. This is a partial list of the projects from where we match the variants. So we have the classical manually reviewed one, and then some large-scale project, including Cosmic, ClinVar, and upcoming the Cancer Genome Atlas. One nice thing is that this UPO-PSI format, there was a poster at ASMS in San Diego last month, first poster that I've seen, as I am aware of the COMET search engine from the transproteomic pipeline that uses this format for variants. So maybe also the Andromeda search engine would like to take a look at this format. It's still in a draft format from the UPO-PSI committee, but basically you have a FASTA file with a large header, and all the informations of variants and PTMs and other sequence features are reported in this very big FASTA header, and then you have the sequence. And in the future, it will accommodate also the proteoform information for the top-down world, okay? So at the moment, we do have this PEF format available as one of the response content types of this API, and we report only variants for the moment, no PTM stuff. We will wait that the format gets 1.0, and then we will start including all the other features. But for variants, it's already there if you want to give it a go. And not only we do report it in this format of the RESTful API, but also on the FTP, and in tabular format, but also for tracks for genome browsers, like BED, BigVet files, and track hubs. So you can visualize the variants and the UniProt feature in your favorite genome browser, like the Ensemble genome browser, UCSC genome browser, maybe others as well. So next steps will be expand the range of repositories, MS proteomics repositories, which we cover, some are reported there. Start including PTM-specific data sets into UniProt, and then make them available all over the place, like in FTP, RESTful API, and on the website in a graphical view. And start adding services to the RESTful API. So knowledge-based services like disease services, enzyme services, and this very nice project in collaboration with Ensemble to develop a variant FF predictor for peptides, okay? This should be very nice. So this is how you access UniProt data. I just want to stress again that we do have a release number version. And for the RESTful API, it's very nice because it dynamically provides examples in CURL, Perl, Python, Ruby, Java, and R for the moment. But upon request, we can add others as well. So acknowledgements for the proteomics are these repositories, which some of them we have already established a collaboration with, some we are just developing it. And this is where the funding comes from for UniProt, also divided by the 3 constituents of the consortium. This is the group, very big. I don't know if you can see the colors, but anyway, the 2 chunks of people here, curators and developers, color-coded maybe, I don't see it from here, but with the 3 constituents of the consortium. And I just want to finish by advertising a tutorial that we will have at the ECCB next September, from PDB and UniProt about programmatic access to protein information. Thank you.
Okay, thank you very much. That was a really nice overview. Thanks again to the rest of our speakers this morning. Maybe we can have time for one question, but then you'll have to find the speakers during the coffee break for additional questions. So raise your hand if you want to be the lucky one.
I had a question to the last talk.
Okay.
The reference proteomes in UniProt do contain tremble entries. Can you speak to how you select those that make it into the reference proteome?
Yes, very good questions. Yes, even the human one has tremble entries. And the main reason is the one that I showed in the sequence data flow. For instance, for human, if there is an ensemble corresponding entry, we create a tremble entry until we have time that curators take a look at it. If there is a corresponding paper to do some manual curation. At that point in time, that tremble entry will become a SwissProt entry, or maybe will be merged, or maybe will be doomed, well, this is not really what we want. So mainly because they are present in the corresponding reference genome. There is no reference proteome without a reference genome, whether the genome is coming from Ensemble or from RefSeq, but that's the case. Yeah.
Thanks once again to our participants' presentations this morning.